Good morning, everyone. Welcome to Concordia University's Forest Space. Thanks for joining us for today's event, part of Planetary, Public and Personal Health, Wellbeing and Justice, a Sustainability Across Disciplines Conference. Today's event, the title, At the Intersection of Behavioral Ecology and Genetics. This is with Sarah Turner and Marie-Julie Favé. We'd like to begin by acknowledging that Concordia University is located on unceded Indigenous lands here in Tiohtià:ke, Montreal. We respect the continued connections with the past, the present, and the future in our ongoing relationships with Indigenous and other peoples within the Montreal community. Here at Force-Space, we work with our university community to help mobilize knowledge, co-creating daily activities that examine research questions, projects in development all across the university. Now, we're running today's event as both a live stream to YouTube and a Zoom meeting. So you're joining us either of those locations and you have comments or questions, put those into the chat and we'll do our best to read those out a little bit later on. Those of you here in the space, if you have any comments or questions, just raise your hand and we'll run a microphone over to you. Okay. With that, it's very much my pleasure to hand our microphone over to Professor of Biology as well as the Principal of the Loyola Center for Diversity and Sustainability and the Co-Director of the Loyola Sustainability Research Center, Dr. James Grant. Welcome back.
Thanks, Doug, for that comprehensive introduction, so I don't have to do so much. And thanks to Force-Space for partnering with us yet again this year for our 9th annual Sustainability Across Disciplines Conference. Yay. Our other partners this year are Future Earth, the School of Health, and our long-term partner, the Department of Geography, Planning, and Environment. So thank you to those people, for those entities, for supporting this conference. Doug, thanks also for that territorial acknowledgement. If you're tuning in from somewhere other than Tiohtià:ke or Montreal, this might be a great time to check out who owns the land, who originally owned the land in which you're situated. And perhaps Rebecca will put something in the chat. Thanks, Rebecca, that will help that. Okay, well, welcome to day 2 of our 5-day conference. We've got 3 exciting events today. If you want to see the complete schedule for today or for the rest of the week, maybe Rebecca will also put the schedule into the chat so you can look that up. So we kick off today with an exciting 2 presentations in a link theme at the intersection of behavioral ecology and genetics. I'll introduce both speakers now and then turn it over. Our first speaker is Dr. Marie-Julie Favé. She's an assistant professor in the Department of Biology at Concordia, where she uses population genetics, evolutionary biology, and artificial intelligence to better understand the origins and development of human diseases. So right on our conference theme. And our second speaker will be Dr. Sarah Turner. She's the co-director of the Loyola Sustainability Research Center and will be the new principal of Loyola College for Diversity and Sustainability. Thank you, Sarah, for taking that over from me. She's also an associate professor in the Department of Geography, Planning, and Environment, and she is an expert in behavioral ecology and the conservation biology of primates. So, all right, so our first speaker is Dr. Favé. Please take it away.
All right, thank you, Jim, for the introduction and the invitation to come to Fort Space for this exciting event. All right, so I'll tell you a little bit what my group at Concordia research, and we'll discuss today from family vacation to human health to little surprise results from wild animals as well by the end. All right, so last summer I visited Vancouver Island with my family. So let's start with just looking at some of these spectacular landscapes that we visited and that we really feel part of it, right? And then on our way back, this is what happened when we were driving back from Tofino to Victoria, where as it's becoming the rule now, a wildfire was raging close to Port Albany. The air was completely muddy and visibility was nil, and that is a recurrent problem every year. And not only from the Western Canada, but it's if you remember, we also got quite a bit of forest fire pollution back in Montreal as well. The first picture on the left was taken at my house in Montreal 3 years ago, where Montreal was elected as the worst city in the world for air quality. And it led to this very impressive picture in New York where the sun was orange and it looked like completely apocalypse landscape, right? And this is not getting any better, right? And it affects everyone, like not just us as humans, but also all our ecosystem and all the organisms that live in those and share those territories with us, or we share with them. And this is the framework of the One Health, where the health of our environments does affect everyone's health, including animals and humans, and our environments. So one thing we've been looking at in my group and previously that I've looked at is, this air pollution is really kind of encompasses the effect of this effect on the ecosystem, but as well of any organisms that will share those environments with us. Just to get a bit more in details, it is largely, defined as any kind of pollution that's in the air. If we go a bit more in details, we often focus on like this particulate matters, PM 2.5, which is what you would actually see on those pictures from the wild forest fires. And our air is often also polluted with ozone, nitrogen dioxide, sulfur dioxide, and many, many other things, such as heavy metals and organic compounds. And it's defined as the 5th leading risk to global health by the World Health Organization. One of the most direct effects on human health that you guys probably felt during those days where the air quality was terrible in Montreal, it really affects directly our respiratory system by turning out inflammatory pathways. It increases the risk of some pulmonary diseases. It exacerbates asthma attacks. It can even cause DNA damage, so directly affecting our own genome, and can lead to other consequences such as on the cardiovascular system. So to get a bit more into the health, and I try to keep that like kind of high level, even if you see a lot of things on the slide, just focus on like the message where, we all have a healthy basis, kind of baseline in our health where all the cells are doing their job kind of on a baseline level. But anytime we have some sort of exposure, whether that's a virus or it's caused by pollution that those small particles are getting into our lungs, whether that's coming from cigarette exposure, well, all those cells are leaving their resting state and going crazy, and they will start trying to manage those stress, and they will start secreting different factors and try to fix the damage. But if your lungs are constantly being exposed to these pollution, it will eventually translate into more chronic problems. So there's always this balance in your lungs between their protection and the overreaction. So one thing my group is doing is trying to study this, balance at different levels. So looking at what we call multi-omics. So we could look at how our genes are being built, but we can look also how our genes are being used, and that's highly dynamic. This can change quite rapidly. And not only how the genes are being used, but also how our epigenome or our proteins are also being dysregulated when exposed to all of these different respiratory irritants. So today we'll look a little bit how pollution can dysregulate this baseline homeostasis level in our immune system, how our personal genetic variation, our personal genetic background can modulate our response to those irritants, and how genetic traces can be used to monitor those environmental exposures. So I've worked in the past on those, there are really large biobanks in multiple countries, including Canada, where the CAN-PAT study, I think I have it here, yeah. So the CAN-PAT study, it's a biobank that has recruited more than 300,000 Canadians across the country. And they have happily given blood samples, health record and measures, so we can study how this environmental exposure eventually affect their health. On the top here, on the top right, you can see what these participants has given us blood, and blood is a fantastic tissue because it really includes a lot of things. It's a messenger that goes around your body every single second of your life and gathers all these messages from the different organs. And what we're going to look at into that is this beige section of the blood that is made of the white blood cells. So that is your immune system, and it's a really diverse tissue. It's composed of many different cell types. All right, so we have this blood sample, and also some of you might know about a really beautiful database that is linked to the CANPAT participants, which is the Canadian Urban Environmental Health Research Consortium, where we have environmental exposure over many, many years that are available for every single of these participants. So the first study that we performed was to look across the Quebec landscape. So there are, we selected participants from 3 different cities. From the CANPAT biobank. And we selected these cities for many reasons, one of them being that they have pretty contrasting environment, with Montreal being a large city, Quebec a smaller city of a smaller size, and then Saguenay, which is up north along the Saguenay River, which is smaller towns, made of smaller towns. However, it is also home to a large aluminum industry. So we recruited, from these participants, we selected 1,000 of them to actually look at, and here don't worry about the very details of the genetics, but what we're gonna look at is how these participant genes are being used by their immune cells that are going around in their blood. So we also have other information of these participants, but we'll start by looking at these RNA sequencing, which means how do these genes actually work? So what you see on the map is where these participants live. And on the little graph here, no needs to understand any of these details, but what you can see here is every single point on the graph is a gene in the population and how it's being used. And the more red you see, the more genes are being used differently across cities that have different environmental exposures. So from a geneticist, when you look at this, I'll give you the clue. This is actually a lot of genes that are being used completely differently between those cities. And by looking at big databases, we can tell that these genes are actually involved in they were associated first with the air composition measure that we collected from the Canute Consortium. They're involved in coagulation pathways and inflammatory pathways. We can also look at big databases of genes function, and we know these genes are actually used in oxygen and gas metabolism and distribution in the body, which lead us to suggest that this is really because of this gradient in air quality that our immune cells in our body are being affected by these exposures. We also see that these participants be exposed to higher level, particularly of SO2 exposure in the Saguenay region. They have a much higher risk for cardiovascular conditions caused by higher arterial stiffness, so how hard your arteries become as you age. And it's also associated with asthma and lung diseases. All right, now that we have discovered this effect of the air pollution in Quebec, we've asked a little further question where those immune cells, they also produce those little factors under inflammatory condition called cytokines. So cytokines are little messengers that macrophages, so this blob immune cell, produce to tell the body something's going on. There is some inflammation going on. I'm being stressed by something that's not good for me. So we also profiled cytokine across 8,000 Canadian, across different provinces, mostly Ontario, Quebec, and the Maritimes. And we asked the question whether we could see similar pattern of air quality or air composition exposure with how much, of different cytokines are being produced by our macrophages. So again, here, don't worry too much about the details of it, but those little graphs, let's just take the one on the left. So every round circle here represented different cytokines. So they all come in different flavors, the cytokines, and we sort of know what each of them actually do in the body. So the higher up you see a circle, it means it's more different across people living in different NO2 exposure. Where, for example, if you see this, the one that I highlighted, which is called IL-1B, so this would be interleukin-1B, it is expressed or present at much higher concentration in people exposed to NO2, which is usually generated by heavy traffic. And this actually is also confirmed what we found in our gene expression study previously. We also find similar pattern for PM2.5 exposures. And we also link that with a big study that we participated in where we do see an overexpression of these particular cytokines, so these messenger in participants that have lung cancer, and especially lung cancer in non-smokers. So people that are exposed to worst level of NO2 tend to have these interleukin being overexpressed in their body. All right, so the next step that we did is to look at the diversity of those immune cells in our body. So they also come in different flavors. There's different cells that are, they each do their different function. And again, so don't worry about all these very details of data analysis, but instead of looking at only, the combined results of all the immune cells together, we focus on particular cell types. So they each have a very specific task to do. Some will react more readily to general environmental exposure, other ones will react to pathogen, and they will learn and remember when they encounter particular exposures. And in our case here, we'll look especially at those SO2 exposure levels that we saw were higher in the Saguenay region. And we do find particular cell type, notably the CD4 cells, which are those cells that actually remembers when they're being exposed to particular pathogens. They help you to train your immune system. And those genes that are highlighted here are particular genes that are being turned on to a much higher level in people that are exposed to SO2. And this might be an indication that, well, we'll get to that a bit later, but this is an indication that there is some pathways that are particularly also known to be involving lung cancer that are being upregulated in people that are exposed to higher level of pollution. All right, so one thing also that my group looks at, and you might also have, this intuition that we don't all react the same way to exposure, right? We have our own genetic background, we have our own, hereditary lineage that will give us particular genetic composition that makes us more or less susceptible to particular diseases. Right, so here's just a little cartoon to show you how two person might be, for one particular genomic position in your DNA, might have a difference. So one on the top has a C, the one in the bottom has a T, and that might give them different susceptibility to diseases. So we can do that looking at the entire genome, right? So for example, we take, lung disease and we calculate how many of these genetic risk factor one person has, and we all put them in a bucket like here. This would be a low genetic risk because the bucket is not full. So whenever you reach the top of the bucket, with your risk factor, it means you will develop the disease. Another person might have its bucket much more filled with genetic risk factor, and that's why we all develop different diseases. However, these buckets are also filled up with environmental exposure. So when we add those purple beads, that's like whenever you smoke, whenever you're exposed to a wildfire, your risk might eventually raise and your bucket will overflow, and that means you will develop a disease. And that you can do that, profile these scores, this genetic risk score across a population where some people have the bucket is pretty empty and some people have a really full bucket and it doesn't take a lot for them to develop a disease. All right, so we had this work performed on 30,000 participants from the Quebec Biobank Cartagena, and that work was led by Dr. Isabelle Gamache in my lab and also fantastic undergrads Samantha Eggers and Alexis Bernier. So they profiled this risk for respiratory function. So that's the FEV1 that you see, which is a measure that we take in hospitals or clinics about how much your lungs are actually working. So the higher your FEV1 is, the better it is. But we all start with different baseline because of these genetic scores that we're born with. So we have a distribution in our population of people from low lung function to high lung function. So I put the little buckets to represent what's the genetic risk factor that each person has. So we have most people are kind of average, but we have few people with like a predicted high lung function, meaning their bucket is roughly empty because they have a very low risk for a lung disease. But we have some people that are born with their buckets that's already full, right? So we wanted to look at how does these people are different from each other. How is this baseline different? So we'll look at how they use their genes differently. And again, let's not worry about like all the data analysis side of things, but one thing that we capture in our analysis is that people with a low genetic risk score versus people with a high genetic risk score, they have a very strong difference in the way their immune system works. So our respiratory function is actually really well combined or trained by our immune system. So depending if you have an immune system that reacts really quickly every time you're exposed to a pathogen or you're exposed to some pollution, well, you might eventually have a better chance at not having a lung disease during your life. All right, so this is just to wrap up this part, that we do see that people with like a high, a very active respiratory system versus people that have, that have a lower lung function from the baseline, from what they were born with, we do see that this is in fact, there's a lot of involvement of your immune system. So people will have a very different antiviral defense system. And this antiviral defense system is also used against any type of respiratory irritant, including smoke and pollution that we breathe every day. All right, this is just a little, in case you're interested in those types of topics, there is also some work in my lab where we eventually look into a little more about how the social determinants of health will interact with our personal genetic variation. Because depending where you're born, where you live, for example, on the island in Montreal, which is depicted in this map on the left, you might be exposed to various levels of air pollution. But also on a more higher level, different neighborhoods of Montreal will include people from different ancestries and different ethnicities. And we're starting to look into that, how our own genetic ancestries, because of the multi-ethnic setup of Montreal, we have people from all over the world, and it is well known that some genetic ancestry might carry different genetic variant that makes them more susceptible to air pollution exposure problems, for example. So that is some work that we're going to pursue in my lab. So if you're interested, please come over to discuss. And then the last part that I wanted to talk about, and I might switch gear a little bit here, and I will link well to the next talk by Sarah, is actually not looking at how our system reacts to pollution, but basically how pollution can actually affect our genome directly, pollution or environmental exposure. So this is a blood vessel where you see the immune cells going around. There's some red blood cells as well, but they don't have DNA. So let's focus on those white blood cells that do have DNA in it. We all know, for example, if you're exposed to UV, this can cause DNA damage, this cause a mutation, and you're stuck with it until the end of your life, basically. So these mutations are basically, they act like a little stamp in your genome. And your genome is like a passport of all your environmental exposure that can cause DNA damage. So this accumulates over time. As you accumulate stamp in your passport, as you travel around the world, your genome will accumulate those stamps in terms of mutations. And we can actually profile those mutations that you acquire during your life. And each of them have a particular, these stamps can tell us where they're coming from, so which exposure has caused them. So that's just an example. For example, if you have a UV or if you have a chemical exposure, this will cause particular DNA damage that we're able to track. This can be seen in lung cancer, where we do see that where people are exposed to higher level of pollution, you have an increasing number of particular specific signatures or particular changes in your DNA, where we can attribute that to an increase in exposure from pollution. And in that case, it was profiled across many countries where those particular mutation can be detected at an increased level as you increase the pollution exposure. Okay, I'm just going to skip this part because I think that's a little too much, but we're kind of trying to profile these mutations across different neighborhoods in Montreal in healthy people to see if we can capture an effect of this air pollution in our genome. But we can do that on not only human, but on various different animals. And some animals are much better, might be better models to understand these effects of environmental exposure on genome because they live across many different environments, they have different genetic compositions. And one animal that we recently turned as a biomonitoring species is this little lovely penguin that lives in the Antarctic. And that seems like a really funny choice to start with. But this is, it was driven by many different considerations. A major one being data availability to do the work, to do the computational work. But these lovely penguins, they also experience extreme environment, lots of UV, cold, oxidative stress. Different colonies of the penguins have very different exposure to pollution. They have a long lifespan, so they accumulate mutation to environmental stress. And also there's a colony in the Montreal biodome that we will be able to study eventually as well. So we selected this species to start with sort of as a pilot species. We adapted the human cancer computational pipeline to profile those stamps into the penguin genomes. And we're trying to identify the possible cause of the acquired mutation in those lovely little Gentoo penguins. So we have our first results on it. This was a massive work by our research associate to actually work on penguin genome. And in this graph, what you see is, well, first, this is where they live, the map. So they live in the South Shetland Islands in the Antarctic Peninsula. There's a bunch of different colonies that are quite differentiated from each other. And what we capture in our somatic mutations, so those mutations that are acquired after exposure, are very two distinct patterns, where some colonies that live more on Anvers Island have a very specific signature in their somatic mutation, and the one that lives in the Antarctic Peninsula have another very different profiles of mutations. So we still don't know what that means exactly. However, this is the first step in our biomonitoring work that we're trying to develop, not only in penguin, but we have some plans, well, first to do it on the penguins that are in captivity as well, to compare with the wild penguins. And then later on, we have some plans to actually work on Japanese macaque. We started this work already. However, I don't have any graphs ready for you today. And we have also some possible work to actually do it also on the Quebec Biobank on 2,000 individuals for which we have high quality data, like actual humans that live across different pollution regime, including, in region where you have high industry level, in the Saguenay region, in Abitibi, where there's lots of smelter and arsenic exposure. But this project just is included in this One Health perspective where we do share our environment with different species, and we can actually adapt very cool computational pipeline to these penguins and macaque to understand better the environmental exposure we all share across our environments. So this work was done by my lovely starting groups at Concordia and also during my postdoc at the Ontario Institute for Cancer Research under the direction of Philip Awadala. And I would like to thank all my students, the funding, and the invitation to be here today.
Thanks so much, Marie-Julie. I think we've got, is there a question in the chat, perhaps? I have questions, but no? No. Well, first of all, thank you for that comprehensive talk. Let's see, environment, social aspects, genetics, and the health of humans and penguins. So that was pretty cool. So is your goal to, I mean, it was so cool that you can link. Do you think you're going to be able to provide a link from exposure to forest fire to smoking? I mean, are these the kind of things you're going to be able to do in terms of a health perspective? Because people can really relate to now what smoking does. And is that what your research will take us to, perhaps?
So these type of inferences, they're being done at the moment in humans for sure. Yeah. Particularly smoking, it's a very well-known exposure. So these signatures that you find, like those stamps, this was derived from a lot of lab work from many different, groups where you expose animals in the lab and you can derive, so those mutations are really like spectrum of different changes using the DNA. So smoking is one that's very well known. I would suspect anything that's kind of close to it, like smoking, forest fire, might resemble that. I think we have to keep pushing the research in that space about different, really profiling those mutations. Like right now, there's about, maybe 90 different signatures that are being described in humans. Most of them we still don't know exactly what's the source. A lot of them are well known. Like smoking aging is one that's very cool to study on a fundamental basis. We accumulate these mutations as we age. So those are usually the ones that come up first. Okay. And then, for some diseases, you have smoking. That's one that's very well known. There's chemotherapy exposure. There's organic exposures. I think there's still a lot of work going on in that space. I have some collaborator, for example, at McGill that work on arsenic exposure. So we're hoping in the lab we can describe also those exposures in different experimental setup. And then once we have an understanding of what the exposure leaves in the genome as a stamp, then we can look into a natural population.
Okay. And maybe titrate the effects of environmental challenges.
Yeah, well, I see that eventually we could use that as a monitoring way of it's non-invasive. You could look at blood, you could look at different tissue as well, because you might not accumulate pollutant in the same way across your body. So I think it's a powerful thing to, this has been highly developed in human and in the cancer space, but I think to bring it to more kind of, we know this happens in all tissue, not just cancer. So to look at these in non-cancer tissue and non-human is also quite powerful.
Cool. Great. Thank you very much for that talk.
Thank you.
Okay, so maybe it's time we should switch to our second speaker, Dr. Sarah Turner.
Yeah, there was just a question. Do we have time? We have time for one more question.
Sure, one more.
Let's do it. Also, Salvador had have their hands up. If they wanted to add a question into the chat as well, we could try to get to that one. But I'm going to read the question coming in from Lisa, who asks, given the data that you have now, could we better evaluate people's risks and predispositions? And if so, could we identify genetic patterns that could warn individuals that they might be beginning to develop the diseases?
That's a big question, but certainly that's a very active field of study where all these large biobanks, so I discussed the CanPAD biobank, but it's relatively small compared to other ones. For example, the UK biobank, which is 500,000 participants that are all genotyped or for which we have genetic information. So we could derive, people have derived personal genetic score for a variety of different traits and diseases. Actually, this big biobank is being used as a training data set to discover those genetic variant for disease susceptibility, and then we can apply it in other biobanks as well. However, I would say that despite all of this work and massive data, those personal genetic score, they remain, for many reasons, underpowered. So it's not a 100% prediction. It's more like much, much lower than that. It can give you an idea if you're at risk. However, it won't tell you if you develop the disease, because we all know our, I wouldn't say our choice, I would say really our life path. Sometimes it's not a personal choice, it's what happened in your life, that you're exposed to different things, to different stressors, and you will develop a disease because your bucket might be full of environmental exposure, and that is not predicted by your genome, right? Also, these models, they look like they can predict some of the risk. However, it works really well on individual of European descent, because that's the massive amount of training data that exists where we do our discoveries. But when we try to transfer those risks into African population, Asian population, then the power of predictability drops by a lot. So there's lots of effort to try to improve representativity of different genetic ancestries for equity in biobank, but it's an ongoing effort that is not resolved by a single lab for sure, not even a single country. So it needs like change in the way we do research, but also there's a lot of barriers in terms of political and legal and data sovereignty reasons as well. But it is something that the community is aware of and is trying to work forward, and hopefully as we train our students to be aware of these equity issues, we'll get to a better place.
Great, thank you. So maybe we'll hold the other questions till the end and move on to Dr. Turner.
Thanks, Jim. And thanks, Marie-Julie, for that wonderful talk. I'm even more excited now to be working together in the upcoming months. I wanted to talk a bit today about behavioral responses to challenging circumstances. Our field research on behavioral plasticity, behavioral flexibility in Japanese macaques and blonde and bearded capuchin monkeys. So I don't know, hang on, if some of you have seen the recent monkey in social media, the proverbial monkey in the room, Punch. Punch is a Japanese macaque who has gone viral recently, sort of won the hearts of the internet. He's a 7-month-old Japanese macaque, and he was abandoned by his mother near birth and was raised by the zookeepers, by people, and was given this stuffed orangutan, which he carries around with him in a very, well, you know, pulls the heartstrings. We all sort of relate to Punch's plight, I think, as he's tried to reintegrate into the zoo group. And it's interesting because this is really the first time that that people are particularly interested in Japanese macaques for me. And so I've had a bunch of news interviews about Punch and why why was he abandoned by his mom, and is this a usual behavior? And the thing is, in the wild and in free-ranging groups, this is not a usual behavior. This is probably there are a variety of stresses for Punch's mother. She may not have known how to mom herself, how to be a mom. And because Japanese macaques learn a lot of those skills in their social group. And there could be other stressors, but the zoo itself is even the best zoo environments are stressful for animals and can cause a lot of behavioral stresses, including occasionally infant abandonment. So it looks like these days Punch is doing better for all of you who are following him and his progress. He's been integrating better into his group and learning how to kind of quote unquote speak Japanese macaque. And if you want to follow a little more of our discussion on his story, some of my students and I wrote this article in The Conversation last week about Punch's situation and our work on Japanese macaques. So, maybe you've heard, but humans are making kind of a mess of many things at a global scale. And environmental change and environments change and animals adapt, and that's not news. Thanks Darwin for that synthesis of heritable variation and differential fitness in the face of changing selection pressures. But now the speed and scale of these changes are really affecting that equation. And while adaptation happens in many circumstances and for many species these days, natural selection can't keep pace with the speed of anthropogenic changes that are occurring. And at least 68% of non-human primate species are threatened with extinction, according to the International Union for Conservation of Nature. And 93% of primates, non-human primates, have declining populations. So, we can say behavioral plasticity rides in like a monkey riding a deer. To help in these mitigate the negative impacts of a lot of challenging circumstances. And behavioral plasticity varies individually and with circumstance, like how necessary it is to be plastic and how possible it is to be plastic, both individually and among species. And there's really an urgent need to quantify some of this variation and to understand what the bounds of the variation are and how much animals are able to use plasticity to accommodate and to modify their behaviors to deal with all of these changing circumstances. So there's a few useful hypotheses that have been discussed to look at plasticity. The necessity drives innovation hypothesis, the idea that animals, when they're pushed, are more likely to create innovative behaviors and new ways of doing things. And that overall, there's the hypothesis that behavioral plasticity reduces extinction risk for species that are more plastic. So there's many relevant questions that emerge from these hypotheses, and I'm just going to talk a little bit about some of the research that students in my lab are doing and that we're working on. So we work in three main study systems. We look at physical impairment in free-ranging Japanese macaques, so same species as Punch the monkey. We work at the Awajishima Monkey Center in Japan, and I'll talk more about that in a minute. We work on bearded capuchin monkeys, a near-threatened species, and a lot of bearded capuchins are in rescue centers and then get released into the wild in northeastern Brazil. And we do this, our lab does this work in collaboration with Dr. Renata Ferreira at the Federal University of Rio Grande do Norte in Natal. And we work with wild blonde capuchin monkeys, which are an endangered species, and they live in the group that we study lives in a small Atlantic forest fragment surrounded by industry and agriculture in Brazil, also with Dr. Ferreira's group. And we have a little interesting side quest that's not a side quest for my PhD student, Ariane Peroni. So we're also doing some work with captive welfare and movement and foraging choices in calatriches, those little, the small neotropical primates. They're kind of squirrel-sized, and in particular with emperor tamarins. So we've been, Ariane and some other students, Aurélie, who will be starting a PhD with Marie-Julie and me in the fall. And they were working at the Biodome in Montreal, and Ariane and Adele will be going to Peru to collect data in the wild this summer as well. So that's really exciting. But back to the Japanese monkeys. So the Wajishima Monkey Center is located here on the side of Awaji Island in the Seto Inland Sea in Japan. It's between Honshu and Shikoku, the main islands. It's a small, relatively small island. You can drive across Awajishima in about, I always forget the exact amount of driving time, an hour-ish. And the Monkey Center is really an amazing place. It's one of my favorite places in the world. And there's a group of about 400 to 500 free-ranging monkeys, and they come to this human cage where people can go in and feed them, and the owners of the Monkey Center provision feed them in a sort of regular, systematic way throughout the year as well. And one of the things that makes the Awajishima group unusual is the high percentage of infants born with malformations of the limbs and digits. So about 17% of infants up until 2007, or 16%, were born with some level of malformation of the limbs and digits. So you see a variety of examples of individuals here with those physical impairments. Little ribbon down in the corner, who you can see doesn't have hands, and then also just above with the babies, there's one, the individual in the middle, who has the absence of the middle digit on one hand. So a range of disabilities can occur from these physical impairments, and although we don't have up-to-date data and there's changes to the population structure that make it more challenging to estimate, but these days with some birth control that's going on in the population, with scientist colleagues in Japan, Keiko Shimizu and the Nobuharas, but we still know that these limb malformations occur in the population. And we know in general there's higher rates in provisioned groups. There are links to organic chlorine pesticides, so this is where also, I mean, I'm curious to know about exposures and genomics in relation to this population. We know from work that was done in the 90s and the 80s that there's no obvious chromosomal or other obvious genetic anomalies. And currently, Awajishima is the only group with a substantial number of disabled individuals. So the main work that I've done in this population over the last number of years has been looking at how individuals respond behaviorally to having physical impairment and disability. How do they adjust their behaviors to compensate? How do mothers raise their infants? How do infants develop compensatory behaviors and learn to, for example, individually appropriate styles of locomotion? And we've looked at, are there signals of impairment in behavioral plasticity and behavioral sequences? In particular, Brogan Stewart has been doing some work with fractal analysis and behavioral sequences to see if there's more subtle kind of indications of impairment associated with them. Oh, hang on, I just did a double click back. So here's a few individuals who we've studied and examples, and you can see we've seen a number of different compensation behaviors over the years. That the mother is holding her infant actively up to nurse in a kind of supported nursing position when an infant is unable to cling. Usually a baby Japanese macaque would cling to its mother, sometimes from right during birth. Like I've watched a few births. I've been lucky enough to see a couple happen over the years. And, you know, in one case, the infant literally climbed its way up its mother as it was being born. So like that clinging is really usually key. But if you don't have hands, of course, as an infant, you can't. But mothers, far from abandoning infants, they compensate and they provide extra care for them and will even carry them, like Teruko, the monkey there, holding the 1-year-old infant Biwa. She's carrying him in a supported carrying ventral position much longer than a mother of a non-disabled infant would usually carry her baby ventrally. And they find individually appropriate ways of locomoting as well. As you can see, these two locomoting monkeys at the bottom corner of the slide. Monmo there is walking on all fours on her elbows, and Ribbon is using the banister as an assist as she walks bipedally, and she's a habitually bipedal monkey, which is, you know, not usual for the usually quadrupedal Japanese macaques, although all of them can walk bipedally. Sometimes most of them do not do that as a habitual form of movement. So this is just a couple of highlights. This is my student Megan Joyce. She's done some research looking at infants and infant development recently and the kind of timelines and behaviors associated with disability in infants. So she had 3 infants who had different forms of physical impairment, 2 with limb malformations, and 1 who our veterinary colleague, Dr. Keiko Shimizu, visually diagnosed as having hydrocephalus. And I was going to say, and she found those same kinds of patterns where the infants had more supported nursing and more supported carrying, the disabled infants did compared to the others. And in particular, the infant who was unable to cling had substantially more supported carrying than the other infants did. And here are some examples of the moms of those infants. Support carrying, ventral carrying, and dorsal carrying their babies, and supported nursing in different, and also lying down nursing. I suspect that there's more lying down nursing for disabled infants as well, but we haven't run those data recently. And we've also kind of looked at these questions through literature reviews recently to look at how unusual is the situation at Owajishima really, and how much do these how much does the intersection of behavioral plasticity and disability cross species? And so Brogan and a number of my other students and I did a survey of the literature, and we looked at, we found almost 3,000 articles and identified 114 studies that fit our predetermined inclusion criteria for behavioral data, along with a visually apparent disability of some sort or physical impairment, and looked at the potential for innovation of novel behaviors and compensatory behaviors. And we found that there was actually quite a lot of evidence of innovation, of extra maternal care, and of compensatory behavior for disability across many, many primate species. And we also found, interestingly, that 60% of the publications connected primate physical impairment and disability to human activities. So that was something that we, that was something that surprised me, how big that number was, the number of examples of ways that primates can be disabled by anthropogenic activities. Everything from vehicle collisions and electrocution from wires to contaminations and environmental exposures. And it really suggests a very entangled relationship among humans and the environment and primate disability. So it's, in some way or another, we are implicated in making these animals be more behaviorally flexible or not, and then the implications for that as well. And recently, we've been working on a review on animal disability and behavioral compensation across many more taxa, and we have 120 papers included in this review that are non-primate related. So this is a variety of taxa from insects and fish to mammals around the world. And we found really that there's many examples across taxa of social inclusion of animals with disabilities. There's sort of this idea, nature red in tooth and claw, that animals with disabilities might be excluded from social groups, might even receive more aggression, and it might always be negative. And that does happen, but is not the whole story. And we're finding that across taxa, and also many examples of behavioral plasticity and innovation described across taxa as well. I'm going to have to speed it up here. So we're also looking at this question of rehabilitation and release, which is helping seeing how we can use the capuchin monkeys in Brazil ability to learn and behave flexibly to acquire foraging skills and kind of make them more able to survive in the wild when they're released. So I have a number of students who are working on these related projects. And as an example, Michaela Gerwing's work on behavioral plasticity and reintroduction. She was part of collecting behavioral data on a number of capuchin monkeys in captivity and looking at their social behaviors, at the degree of stereotypic kind of repetitive behaviors that emerge from stress and captivity, and then also looking at the picture of the soft release enclosure here, where there were 3 capuchin monkeys who were released into the wild. And of those 3, we have evidence that at least 2 of them integrated into the wild group. We're also looking at space use and foraging strategies in the wild blonde capuchins. So, this is the group that's in a very small forest fragment and surrounded by agriculture and human activities. So we've looked at how they're adjusting their use of space and their foraging behavior to accommodate this small habitat. And Italo Pereira, one of my PhD students, recently submitted this study that's looking at the kind of change in space use from this group over time, using GPS points to look at where the core of the group is using, and finding that they really are changing their use of space with the over time to accommodate particularly arthropod availability, which is actually a really critical food source for them, more so even than the fruit availability. It predicted where they were using space. So just to say that data on how animals adjust their behaviors to compensate for challenging circumstances is needed to understand and mitigate negative impacts of human-induced environmental change. And I'm also really excited to be looking at how environmental exposure to the genome and maybe some of these species might help us with their conservation and perpetuating their persistence in the environment over time. So lots of thanks to our collaborators and funding agencies, and to all of the students, and Jim and Rebecca for inviting me. And thanks especially to Renata, without whom the work in Brazil wouldn't be happening, and to Yamada-san in the top next to Brogan there, and the Nobuharas who help collaborate and facilitate our work in Japan. So thanks very much.
Thanks so much, Sarah. That was incredible. Are there questions in the chat? I thought I saw some coming up.
Yes, if anyone else has any questions in the chat, they can pop them in. We can read them out. Do we have a question in the chat here? Just opening it up.
Maybe there are comments.
Not at the moment. Sorry, Rebecca, are you seeing a question? Yes, there were some questions from usually.
Okay, well, I have a question for Sarah. So what do macaques have to teach us about, integrating, relating to disabled individuals? Are there lessons there for us?
I mean, it can always be a bit dangerous to take some sort of, moral lessons or something from animal groups or species, because there's always an example of something to illustrate.
Yep.
But I will say that I've watched at Awajishima as even very, very extensively physically disabled individuals are treated non-differentially. There's no, there are some differential treatments, but overall they live very similar lives to all of the other monkeys in the group. And I think, there's a whole conversation around disability, and sometimes, disability activists will point out all the many, many ways that our society does not treat people with disabilities fairly or reasonably. And so if there is a lesson, I think, like here we see these monkeys are like, yeah, meh. The baby doesn't have hands. Well, I carry it. Yeah.
They figure it out.
They figure it out, and they don't. The individuals with disabilities find ways of like grooming, of moving, of climbing trees, of doing things that I think sometimes we might look at a picture of an animal without hands and feet and make judgments about, they wouldn't be able to climb a tree or do, and they do. They climb the trees, they run the fences, they run faster than me in the forest. So, yeah.
It's great, thank you. I know
Jim, there is a question that just came in for Sarah on Zoom. And I'll read it out. It's kind of a compound question, but from Lisa asks, why were these species chosen to discuss? Was it because of noticeable, measurable changes occurring that were linked to human behavior? And then, what are your thoughts on more human interventions to counter human-caused issues?
Yeah, I mean, in terms of the species, I've been working in Japan since I went as an undergraduate in 1999 at the Awajishima Monkey Center. So I work with this group, and that's kind of why Japanese monkeys. But and then a more recent collaboration with Dr. Renata Ferreira has led to this really lovely access for our lab and collaboration with her lab for students in Brazil, working in Brazil. And it was an opportunity working in Brazil with Renata to incorporate more conservation and kind of like landscape questions in more spatial questions with fragmented habitat in a wild population. And then, looking at the individuals in rescue centers, there's lots of overlaps in terms of welfare and ability to learn. So it was kind of a nice synergy, and these are, I don't know, things have a way of falling into place. I guess these are the places that my lab has been working for the last while. And the second part of the question, yeah, I mean, I think we need to be doing more to mitigate the negative effects we're having. I mean, and some of that is direct interventions like rehabilitation and release of animals that were formerly pets. And I have a number of students working in rehabilitation and release contexts in Brazil, and that's kind of a hands-on way to make a small difference and give individual monkeys a chance at a wildlife who wouldn't have had it. I think that needs to be coupled with the kind of conservation planning and management and working with local communities to really have conservation of the entire ecosystem and all of the biodiversity involved. I mean, one of the wonderful things about these monkeys is they can be flagship species for entire forest ecosystems where, it's really critical that we have conservation of the entire forest. A monkey like Punch can sometimes give us the impression that animals don't require their whole ecosystem because they're living in a zoo and you just see their zoo environment. But actually the wild species, of course, require a whole ecosystem to live in. So.
Great. Time for one more? Or
We don't have another question. We don't have another question at the moment for Sarah. We did have one for Marie-Julie from before, if you wanted to return to that. Do you want to try?
Emma was asking, at what point do we decide that something is harmful enough to warrant legislation to regulate it? And can this physiological data provide the appropriate information to drive regulation?
I believe this is for humans, right?
Yes.
I think this is an ongoing question for many different cities and different governments. I mean, I'm not too skilled into that space exactly. I am involved in I would say one example that's been in the news a lot is the smelter in Rouyn-Noranda in Abitibi, where it is an economic driver of the region. However, it does emit a lot of heavy metal exposure to the population there. There's currently a call for some money for scientists to study the effect on health of the consequences of these exposures. And there's a lot of political issues where you do want to keep the economic driver for a particular remote region because that is, what makes people living there. And the first determinant of health is your income, right? So if you lose your income, your health's not going to get better either. However, could we do it with better regulations so people can mitigate the effect of the pollution? So that's a big question. From the last decades of public health work, whenever scientists get into understanding better exposures, there's been, for example, lead exposure. We thought it was fine 50 years ago, and then we realized it actually affects a lot of the health of people being exposed to it. So then regulation, after a lot of fighting, came in, and then things are being lowered. And it will keep, medical professional will push to keep trying to lower these exposure because it is, affecting people who work there and they think they're fine, but their kids are being exposed when they go outside. Their domestic animals are being exposed when they go on a walk with their dogs. I do think having the most evidence we can might help us introduce at least mitigation strategies for the population living there. And that's hopefully the goal, at least for IBTB. That's where, there's a lot of politics involved, but this is where hopefully some of our communities can play a role in at least understanding what are the risks and how we could mitigate them.
That's great. Well, maybe on that hopeful note about science and society, for society, we should wrap this up. So thank you very much, Marie-Julie and Sarah. What a great start to the day.
Thanks, Jim. Thanks, Sarah. Thanks, Marie-Julie. Just wondering, and also thanks for everyone joining us online, also for your questions. It was great to have that. We're going to be closing up the Zoom and the live stream now, but this does remain available on our YouTube channel, and you can revisit it there. You can share it, and we're going to reset for another event coming up at noon. Issues in environmental science, health, biodiversity, justice, and waste. So come join us back for that. Thanks, everyone.